Before we begin today's session, I would like to take a moment to introduce CRISPR Biosystems Private Limited. It is established in 2003 and headquartered in Mumbai. CRISPR Biosystems is one of the India's leading life science companies, serving researchers across the biotechnology, pharmaceutical, biopharmaceutical, vaccine, CRO, and academic sectors. Over the past 2 decades, CRISPR has built strong expertise in immunology, cell biology, molecular biology, and cell and gene therapy, also translational research. Through partnerships with more than 50 leading global biotechnology companies, we provide Indian researchers with access to cutting edge technologies, reagents, instruments, and research tools across a wide range of applications. Since 2008, CRISPR has also been manufacturing its own portfolio of ELISA kits and immunoassays, including cytokine assays, monoclonal antibody assays, and other specialized solutions for life science research. And now, a brief introduction about myself. I am Dr. Anisha Polle Shikdar, Product Manager for Molecular Biology and Cell and Gene Therapy, CGT, at CRISPR. I hold a PhD in cardiovascular biology and I have a few years of industry experience. At CRISPR, I manage the molecular biology and CGT portfolio, including starting materials, viral vectors, and related molecular biology products with a focus on advanced therapeutics. I would also like to introduce our webinar partner, Applied Biological Materials, that is ABM. So ABM is a Canadian biotechnology company headquartered in Richmond, BC, with over 15 years of experience delivering quality research reagents and custom services to life science scientists worldwide. ABM offers one of the most comprehensive portfolios in the industry, spanning PCR enzymes, antibodies, viral vectors, lentivirus, AAV, adenovirus, CRISPR tools, cell lines, and RNAi solutions, all under one roof. ABM combines catalog-ready products with highly customizable services, including stable cell line generation, virus packaging, gene synthesis, and CRISPR knockout, making it a true end-to-end research partner. With a commitment to affordability without compromising quality, ABM is the go-to supplier for academic and industrial researchers who need reliable, innovative tools to accelerate their science. With that, it is my pleasure to introduce today's speaker. We have Dr. Miguel Mantilla, Head of Business Development at Applied Biological Materials ABM, with us today to share his expertise on viral vectors and CRISPR for building disease models. So, Miguel Mantilla, MBA, PhD, is a biotechnology executive specializing in translational life sciences, gene modulation technologies, and advanced cellular engineering. He is currently serves as Head of Business Development, EMEA, at Applied Biological Materials, where he leads strategic expansion across Europe, the Middle East, and Latin America, focusing on the deployment of CRISPR-based systems, viral vector platforms, and engineered cell models into academic and biopharma research environments. He holds an MBA from Esden Business School and MSc in Molecular Biology from the University of Buenos Aires and a PhD in Neuroscience from the Wisman Institute of Science, with a research background rooted in neurobiology, systems neuroscience, and cellular signaling. His work integrates molecular and cellular neuroscience with industrial biotechnology, with emphasis on RNA biology, gene regulation, and platform technologies, enabling functional disease modeling and translational research. I kindly request all participants to remain on mute throughout the presentation. If you have any question, please submit them in the chat box and we will address them during the Q&A session. With that, I will hand over the session Dr. Miguel for today's presentation. Over to you, Dr. Megan.
All right, good afternoon, everyone. And there I am. Good afternoon, everyone. Thank you, Anisha, for this very kind introduction. I will just like to give a little bit of the update because we don't have 15 years working in the market. We have been working for more than 27 years right now. So as Anisha mentioned, I am right now based in Spain. This is where my home is. And I support directly the European Union, India, and the Middle East. I'm very happy to come back again to speak to an Indian audience. It's been a while since I've been in India. The last time that I was enjoying a very nice kingfisher in Goa, and then I went to Mumbai for some visits. With that in mind, I'm going to start sharing my slides. So please, Anisha, let me know when everyone is able to see my presentation. Like Anisha mentioned, we're going to have a Q&As at the end, so feel free to shoot any question that you may have. I'll be more than happy to address most of them at least. And like Anisha mentioned, at ABM, we're committed to bring translational research into your laboratories. And for that, we have been working in collaboration with Christian. We took a while to choose the right partner in India because India is a market that I love in terms that you are not willing to commit on quality. And that's something that ABM is bringing to your desks and benches because as we are working across the globe, we're going to share a little bit of the people that have been trusting in our technology. We're committed to work towards the development in science. We're very happy to move into endeavors in which we know the advice that we can give is going to be very straightforward because we have an experience of almost 30 years in transforming something close to 2,500 different cell lines. And that's something that we feel very proud of. But we're also very happy to collaborate in ventures in which we don't know exactly where we're going because that's basically what science is. Just to give you one example, we're working with a company in Texas called Colossal Bioscience. If you look a little bit in Google, you're going to see them that they are very striking in the news because they are doing a very polemic project as trying to bring to life woolly mammoths. So as you can imagine, there are not so many experts in the world in mammoth cloning, but we're very happy to collaborate in these kind of endeavors because we're risk endeavor, we're willing to take challenges and work them in collaboration with you. So Anisha briefed out a little bit of what we do, but I have a slide summarizing a little bit of the expertise that we have in different points in the workflow in transforming cells, especially using CRISPR technology. And in that way, we can support you in the whole workflow because, as she mentioned, we do the whole custom service if required, or we can just sit with you at any point in the transformation, in the verification, in designing the right targets, in choosing the right transformation or transduction techniques to be working on this. Given the expertise that we have, and because right now the World Cup is very hot because Canada is one of the hosts of the World Cup, and for us being a Canadian company and me living in Spain as a Spanish guy, football is very important. So I am very proud to say that we collaborate with the Cristiano Ronaldos and Lionel Messi's around the world in science. So companies like J&J, Astellas, Abbey, Roche, Sanofi, they are working together with us in several projects across the globe. And this is just to give you one example. If I go into academia, we're going to find also very important names such as Cambridge University, the Weizmann Institute in Israel, where I took my PhD, and so on. So the expertise that we have is very happy to bring the collaboration that we're expecting to bring to your labs because we know as people that came from the scientific background, what kind of challenges you're facing. And something that I really appreciate about the work that I have is that, like Anisha mentioned, I'm working in 3 different geographies, and those geographies are facing so way different challenges. So as I collaborate with the European Union, as you can imagine, the budgets are a little bit more generous than usual. But when I cross different geographies and I start talking to researchers in Latin America, in India. I know that the funding is not that generous because your governments are basically taking different decisions, unfortunately. So that's something in which the collaboration that we have with Christian has fed us a lot into what kind of support we need to give you, not to overlook the science, but also, and more importantly, into bringing the direct technology that we have. So I'll start jumping into science because this is why everyone is here. So for CRISPR application and challenges, we have a lot to dig in. And more importantly, when we work with CRISPR, one of the main challenges that we're having is that right now we have people with some great expertise and people that are right now venturing into understanding basically even what CRISPR is. So for example, when I speak to people around the globe and I ask them, what is CRISPR, for example, and we're all scientists, so we're normally capable to categorize what something is and where it can stand in the taxonomy way. So, for example, if I ask what is a reptile, you're going to say that is a vertebrate. If I ask what is a frog, you're going to tell me, of course, that is an amphibian. If I ask what is DNA, you're going to mention that, of course, is a kind of molecule called nucleic acid. But when I speak about CRISPR, people know the application, so they know that it's a gene editing tool, but they cannot point out what it is. So this is where we're going to start, just to give you a context. When I came to Malaga, I was very excited because I was living in Canada until September last year. So I relocated in Spain so I can be closer to everyone. And I was very excited because I am Spanish and I was looking forward to get Spanish food. So I live in Malaga and they told me, can I find the Spanish food in Malaga? But no, apparently we're having a boost and a bloom of Indian restaurants here. And that's something that I love because coming back from Canada, I have a lot of friends from India and I love your cuisine. So I'm very happy that because of the British influence that we have here, there is a lot of Indian restaurants. And something good that they have is that they know what kind of food they give to someone. So if they notice that you're not from India, they're going to give you a decent level of spice because they know that you cannot handle it. But when they have an Indian person coming, they give the right level of spice because they know what you can handle. So being said that I'm talking about food, CRISPR is a little bit like a khichdi. I don't know how to pronounce it. Sorry, it's a dish that I learned in Canada with some Indian friends, and they told me that is some food that you have in different versions across the country, and it's basically made of leftovers. And that's basically what CRISPR is in science. So CRISPR has become well known since 12 years ago, but CRISPR is almost as old as I am. So CRISPR was discovered in 1985 because they were trying to take a little bit of the metabolism of bacteria, and they found these CRISPR molecules and they baptized it as CRISPR. But just recently, they are starting to take the leftovers of science and they prepared this beautiful khichdi that now we are working right now. And this is something that always reminds me a quote that I saw in my institute where Mr. Federico Leloir, or Dr. Federico Leloir, studied as well many years ago. He's a Nobel Prize laureate, and they have a quote that is in Spanish, but I'm going to translate it for you. It basically, he's describing that science is like a dark room, and scientists are just people putting their hands in this dark room and trying to grab something and make sense of what they grabbed from this dark room. So many discoveries are basically scattered, and in some places where we have more communications or where we can discuss a little bit more about different findings, we can give them some sense. So that's the beauty about science, and I'm very excited to be working in biotechnology and biopharma in 2026 because right now we have a global communication where publications in India can be well known and quickly acknowledged in different places such as the UK, Germany, Japan. So the communications between scientists, even though you're not talking over the phone with people, is active. Basically, the publications are something amazing because people can talk and make science come to life. So the difference that we have for CRISPR is that in vivo application and in vitro applications can become a little bit more challenging depending on the cell models that you're working with right now. So in that way, we can establish different methods that we have been tailoring in the past years. So as I said before, we can give you a straightforward advance. And that advance is going to give you a little bit of savings into something that normally people do not appreciate too much, which is time. Time right now is a very precious asset. Because yes, you can have something in a proper price, but you may spend a couple of months trying to standardize a technique. So that's something in which ABM ventures a lot, is in giving you a straightforward advice. Let's say that you want to use some given lentivector in a HeLa cell or in a neuroglia cell, so you can have a direct advice and a direct approach into the different 4 stages in CRISPR technology. So if we summarize CRISPR technology, we basically can put it in 4 different steps. So one is gene editing and cloning, in which we need to design the sgRNA target, choose the right approach to work with some given technique, then select the proper gene delivery. So you're going to start some very interesting questions like, am I going to use a lentivector or am I going to use a lentivirus? Because in that way, you can have some pros and cons in analyzing what is the best way to approach into that technique. Finally, what you're trying to achieve at the end is cell engineering. We're going to be engineering some cells, and that's something that the clone selection takes a little bit, and for us, clone selection is an art. So we have very well expertise scientists back in Vancouver, where the headquarters are, in which they're going to be selecting and improving constantly this kind of monoclonal selections, because they are very well specialized into doing that. But finally, you always have to come across with the validation. A CRISPR technique without the validation is basically saying that you've made something nice, but don't bring flowers to your anniversary because you basically forgot. So the conversation with us can start at any point in time. Can start from the very beginning, in which you can tell us what is your goal. And in that goal, we can actually give some approach and basically, as I said, the expertise. And this is what I put the logos of pharma. The experience that we have in pharma is something that we feel very privileged to hold, because it's different when you support an academic customer, because at the end, the progress and advance that you develop in academia eventually will turn into a publication. But it's not the same case in biopharma, because in pharma, everything that is working, basically, you're not going to see it published because it's under secrecy. But as we collaborate with so many pharmaceutical companies in so way different projects and contexts, we know what we learned, and in that way, we can give you that kind of knowledge base. So basically, the features that we can have for the lentiviruses and vectors that we do are very variable, because we have been improving the delivery, the efficiency of the vectors and viruses that we work with by removing several viral proteins that are not really relevant. And in that way, we can give a little bit more room for the lentivector to hold better the different sequences that you actually needed to have. And basically, we have learned from the 2,500 different cell lines that we have engineered that there are different MOIs that we can work with, so we can be more efficient into improving the delivery and the transformation of these cell lines into what we're expecting to become. So as I said from the start of my talk, we can give you a straightforward advice, and that's something that we have available. So if you're working, for example, with any of these very common cancer cell lines, don't take months trying to find the right MOI, because you can just give us a call and we can share with you these different charts that we have about what is the best MOI that you can go straightforward, basically given some viral titer that you're going to be working with. And in that way, you can enhance not only the price in which you're going to be acquiring some material, but you're also going to get a straightforward progress into your lab. And that's something that people don't actually measure. Also from the AAV perspective, I just want to say that right now, especially when people are trying to jump more into in vivo models, they are using more and more AAVs compared to lentiviruses and lentiviruses. Basically, the applications, the advantages, and especially on the therapeutical approach, is better to have something that comes to the body or to the living organisms in which is supposed to do something and then eventually fade away. And that's an advantage of using AAVs in contrast to lentivectors or lentiviruses. Also, it's worth asking if you want to have a system that you can control or that is going to be active the whole time. So that way we can also suggest in some cases to use inducible vectors. Basically, the inducible vector is going to give you a little bit of control by turning this. Also, in some cases, probably depending on the theoretical approach or the nature of the gene that you want to knock out, it might be that you're not looking for a full knockout. It might be that you're looking for a knockdown, because in that way you can have more control and more managing variables that you can see on your system and actually perform more assays. So in complement to the sgRNA library that we have, we also have our miRNA libraries based in overexpression, in inhibition, and so on. And I wanted to bring something new because right now I see that more and more people are seeing that CRISPR is very attractive, but it also can be a little bit limiting, because basically for grown people, you cannot target an application in which the whole genome or the whole target sequences that you want to modify is going to be modified in the thousands of millions of cells that we have in the body. So as people know, CRISPR grew tremendously in publications in the past 10 years, and this is what is happening right now with circular RNA. So circular RNA basically is made of different leftovers because it was discovered decades ago, but right now it's taking some pace and some interest in more and more people. So basically, like the name states, circular RNA is a RNA molecule in a circular stage, and is capable to work as a sponge. So the advantage of circular RNA is that you're going to have a larger sequence, because normally circular RNAs are ranging between 600 to 900 base pairs. So you can do a lot. You can design a circular RNA to be as a sponge for a small peptides, for proteins, epitopes, and for some mRNAs that are there being expressed. So you can do a lot of circular RNA. So in case that you're interested, just give us a call, because we're going to be more than happy to give you a little bit more of an overview of this. And also it's very interesting for me, as I work with different labs, that CRISPR is not the only gene editing tool, but is right now the hottest one, basically because it's a way more straightforward approach compared, for example, with zinc fingers. I don't know if any of you have suffered designing zinc finger approach, because basically zinc fingers, you take thousands of designs, and if you're lucky, one of them is going to work. And basically, the complex nature of zinc fingers is going to be very scary for some people. So in that way, I understand why some people is getting away of that. Okay, now touching base, because I want to move on into CRISPR technology, I promise to give you some definitions. So as I said, many people know what is the function and the potential that CRISPR may have. So yes, it is a gene editing tool, but the main question that we should ask is what it is. So basically, the name of CRISPR in our definitions in science is a type 5 restriction enzyme. So it's basically very similar to ECOR1 or HINT3 or this different restriction enzyme that people are using in the lab for years. The only part that is missing is that you need a guide RNA that is going to allow the Cas9 complex to attach to a little piece of DNA. So basically, if we give some example, the Cas9 is going to work as a knife because at the end restrictions enzyme are going to cut the DNA, and the sgRNA that we design is going to be the hand. In that way, you're going to cut and chop the onion and not cut your finger. That's basically a very simplified approach of definitions of what CRISPR is. So again, it's a type 5 restriction enzyme that works in different applications. And in this process, we're going to be checking on what can be done in the different steps in CRISPR technology. So one of the different questions that people may ask is, what kind of vehicle am I going to use for my sgRNA targeted approach and for also inducing the Cas9 and the reporter that is normally required for this kind of cells? So basically, the main targets that we have are going to be put within a lentivector, an adenovector, an AAV, or a retrovirus vector. They have all different pros and cons. So basically, one of the main things that you need to ask if it's the vector going to fit into your packaging capacity. For sgRNA, normally that's not a problem because the sgRNA is not a big sequence. It's not like inserting a complete gene. But then you have to check what is the efficiency of one against the other. So as you can see, adenoviruses are normally the better or the best ones in having higher efficiency. But also you need to ask if you want a stable integration of your Cas9 and the different applications that you want in your cell, or if you just want to have a transient approach and then letting the system just to fade away. So that's basically what you have to ponder in the first stage of your design. That's something that you need to ask. And again, this is something which we can give you a better understanding. And also to enhance the ability of working with different kind of type of vectors, we have also came across the possibility of not only using viral promoters, because viral promoters are not 100% loyal to the in vivo phase. So being considered that, we have designed a lot of different Cas9 expressing cell lines that are going to be optimizing the CRISPR workflow. So as you can see, we have different types. We have a lot of cancer cell lines. We have also different kind of immortalized cells expressing Cas9, because in that way, we can really have more and more diverse tools into venturing into the different applications that may take place into your labs. The validation, normally, it's also very demanding. So normally, the surveyor assay has been used for a while. And we have also protocols based on surveyor assay. But as you can imagine, right now, we're working with something close to 700 projects in simultaneous. So we are not willing to spend so much money if we can just cut whatever you can have. So basically in surveyor assay, you're going to have a lot of prospects that need to be sequenced so you can confirm which clones were edited and in what way. So you can see in surveyor assay different clones that were selected based on as a partially knockout or just one allele mutated. In that way, you wouldn't know until you actually go to sequencing. But with the Cas9 cleavage assay that we have designed, basically what we're going to remove is all these noise variables of mutated heterozygous CRISPR clones and only work straightforward with those that have the double knockout. So it's a wonderful assay because we have reduced the sequencing budget that we had by 75%. So this is a small kit that I always love to promote because people, I know that you are facing right now some constraints in funding, or you might be just willing to be more efficient. And both ways are, of course, very important to consider. And for the different applications, I'm just going to skip this part of a slide that you have already seen. So for the consultation, you can also let us know if your project or this different sort of experiment that you have in mind is feasible. Because in some cases, some people is just going to see the first look that literature is going to give you, but not really look into the applications. So this is something in which the consultation that you give us is going to probably turn into a very interesting project like this, a very interesting knockout in which you're going to have full disclosure of the sequencing information that we have, so we can actually confirm that the novel knockout has been achieved and in what way, because normally targeting some very delicate gene because they might be lethal can be a little bit overwhelming for some people unless that you know how to approach this kind of technologies, or you know the way in the way we're supposed to work with this kind of cells. And about the different transduction protocol that we have, I'm also very happy to let you know that some difficult colonies or difficult forming cells are going to be also highly approachable for us based on the obstacles that we know how to overpass or surpass. Because as the experience that we have known already not only what is the MOI, but it's also what is the right approach. So in some cases, we have selected different approach into developing these kind of clusters, but also working with difficult to transduce cells such as HeLa cells or HL60 cells. So HL60 are very demanding basically because they have a narrow window for transformation and drug selection is not always so efficient. So in case that you have this kind of approach, let us know because we have a lot of tools that you can take advantage, and I'm going to give you a present at the end. So don't worry that you're not going to leave the room hands-free. But more importantly, you can just let us know where are you having your issues and where are you facing more and more issues in your lab so you can have a direct solution. Or you can just let us know, look, I'm not interested into working with my hands in CRISPR technology. I just don't want to have the ready-to-use solutions. So we also have different kind of genetically engineered cells. So we have developed mast cells using for immunotherapy. We have also skeletal cell, bone marrow, and we're also crossing species. So something that I didn't mention at the beginning is that we are a mammalian company, let's say, or vertebrate company, because most of the genetically engineered cells that we have been working with are basically human or different mammalians, but we also have some fish, reptiles, and snakes. So the collection is growing tremendously, and not only in unique cells, but also in genetically engineered cells. This is one slide that is going to show you the inheritance that we have by developing projects for pharmaceutical companies. So for example, this PDL1 knockout that you see in the right column, it's something that we developed for Merck, because when they were developing the Keytruda assays to test the different efficiency of this interesting molecule for lung cancer therapy, we developed this clone that right now is available for everyone to acquire, not a pharmaceutical price, but also in a very convenient transfer price for you. So this is a little bit of a summary. I'm just going to jump in the last page of the workflow, because as we have been working in immortalization, in CRISPR technology and stable cell generation for more than 2 decades, but it's almost 3 right now, we have been blessed by being acknowledged. This is a Science magazine publication in which they highlight that ABM is one of the global pioneers in this kind of technology. And thanks to this application, we have right now more than 900 different cell lines in catalog, only coming from human source. So you're going to see that some of them are very challenging to work with, but they are there. And they are also capable to perform different kind of assays. So like Anisha mentioned at the beginning of the introduction, the potential is limitless because it's not only what we have right now, but we can potentially do with the different models that we have in our catalog. And coming back, right now we're working with more than 500 projects in simultaneous. So that's something that in most cases is worth considering when you don't have the trained people in your lab, because getting people to be properly trained in CRISPR technology takes a little bit. And if you can trust your technology or your applications into a company that already has a workflow, so for us it's very easy just to put one more cell line in the workflow and keep the collaboration ongoing and keep you updated on the different challenges that we're having in some of the applications. Okay, so summarizing, thank you for the introduction, and I'm just going to finish with a beautiful present that we have, because ABM has been founded as a cell engineering company, but it's founded by scientists. So we're all scientists, and we know what is the different kind of challenges that we have. So in my experience, there are three different kind of labs that I'm facing when I visit people live. So one of them is the expert. In the expert is people that they know very well the technology. They know in some cases a little bit more than we do for some applications. And the second one is laboratories that they are very well experienced in their application, but not in the technology. So for example, we collaborate with neuroscientists like myself or with oncologists that they want to work in oncology, not in CRISPR. So we collaborate with that. And third, we work with people that have read and they know that it's something that they should be starting to be working right now, like AI. So we give them free tutorials. So the present that I have is basically, before I jump into the Q's and A's, is free learning resources for everyone. They are in our website. And this is an initiative that we started years ago, basically because we wanted to have something to give away for people. As, for example, I studied in college, my undergrad, when I was in 2001. And I have seen the same textbooks right now in new editions, but they don't have, for example, a chapter for CRISPR. So if you're free and you have nothing to do in summer, you're going to have a whole curriculum free for everyone in our website in this address. So feel free to jump. We're very happy that Google Ads is showing that more and more students from different nationalities are using these resources for being literate into different kind of applications. So you're going to see in there a tutorial for CRISPR. You're going to see that we have case studies, handbooks, research tools, and in that way, you're going to take advantage of something that is not there available for everyone. So with that, I want to thank you everyone for paying attention to me today. I'm very blessed to be here with you. And Anisha is going to get the mic right now to pass to some Q's and A. So thank you everyone again for your time. Okay, I'm not sure what's happening.
Yeah.
Okay,
Yeah.
there she is.
Thank you. Thank you, Dr. Miguel, for such an engaging and insightful presentation. And we truly appreciate your sharing your expertise and valuable perspectives on the use of viral vectors and CRISPR and the technologies for developing advanced disease models. The session provided a comprehensive understanding of gene editing workflows, viral delivery systems, engineered cell models, and their applications in translational research, from in vitro studies to in vivo validation. Thank you, everyone who joined us today. We are grateful for your participation and hope you found the session informative and valuable. We appreciate you taking the time out of your busy schedules to be with us. Your engagement and thoughtful questions contributed greatly to the success of the webinar. So, the forum is open for the Q&A session.
Hello, am
Yes,
I audible?
yes, Bhupendra. Hello.
Hello, hello, hello.
Hello. Yes,
Yes, am
Shodipto,
I
right?
audible?
Yeah,
Yes.
yeah, you're audible. Please continue.
Yeah, yeah, we can hear you.
Actually, I want to use CRISPR technology for yeah, engineering of bacterial system, E. coli
Okay.
system. Is there any thing available from your company
ABM.
or
Yes, yes.
and I also want to use Leishmania um,
Leishmania.
Mhm.
Okay.
in Leishmania also.
Okay.
Okay,
Okay.
thank you for your question. That's something that I mentioned a little bit at the end. So this is the kryptonite of my company because we don't have right now the workflow for bacteria. So as I said, we're working in cell engineering for mammalian or eukaryotic cells, but mostly for animals, including humans, of course. But right now, the only project that we have been running for different labs is developing the vector that can be used in different systems, such as agrobacterium and different kind of bacteria, but not directly, like I said, for all the human application that we do have. So I regret to inform you that right now we don't have anything for bacteria. If it comes available, you'll be the first person that I will call.
Thank you,
Okay.
Shodipto, for
Thank,
question. Yeah.
thank.
Hello,
Yes.
hello.
Hello?
Yes, Mr. Kishan.
Uh, uh,
Please continue.
yes, yeah, sir. I have a question that what are the accuracy when we use the CRISPR-Cas? Like we have different vectors, and we've used. So which vectors should we use for more accurate accuracy?
Okay, I'd like to return a little bit the question because I would like to understand in what cell model first are you trying to do CRISPR?
Sir, I want to use in vitro and in vivo also. I have MAP cell line, mouse embryo fibroblast. So I will use in
Mm-hmm.
the in these cell lines and also in some other cell lines like HeLa also. So firstly, I want to use in MAPS. So how can I use, means which vector I should use for more accuracy?
Okay, we have an amazing vector for that. It's a backbone that has been already used in this kind of cell models. And it's what we call the all-in-one lentivector. So normally what we're trying also to enhance in the workflow is not establishing three different sets in time in which you're going to be transducing the Cas9 expression, then the reporter, and then the sgRNAs. So normally it's an all-inclusive vector that is going to include in different promoters the sgRNA that is sequenced or is designed and sequenced by us. But if you have your own sequences, we can do it as well. And in a different promoter is going to express simultaneously the Cas9 and the reporter. So for this kind of models, is not in the slide that I had at the beginning. Sorry for that, but we do have some MOI and different transduction mechanisms that we can advise you. Also, something that we recommend using is a proper enhancer. So we do have a proper enhancer that it's not just going to allow the cell to take
Sir,
the genetic material,
okay.
allow the virus to come in, but also take care of the health of the membrane. Because in most cases, for example, when people use lipofectamine, the effect can be cytotoxic. And it's not because of the technology, it's because of the enhancers that you're using. So we do have some enhancers that while they use reagents like lipofectamine, they also include some buffers that maintain the health of your cells. And in that way, you can have a higher viability at the end.
Okay, sir, but I want to ask the vector, like I am using the PX vector, like PX458 vector and the other. So the vector, miss, on the basis of the cell line, are there different vectors which should be used, or we can use one vector for different cell lines?
You can use one vector for different cell lines, but you will need to check on the standardization and transduction conditions for each cell line. In some cases, the conditions can be transferable, but if you can send us an email, I can give you the straightforward transduction conditions or transfection conditions in case you're using either lentivector or lentiviruses for using the same vector for a different cell. So that's something that we have already standardized. So again, if you can just give us an email, I'll be more than happy to give you a straightforward advice on how to work with this type of vectors.
Okay, sir. I will try to mail you and tell all my conditions. Okay, sir.
Okay, terrific. Thank you.
Okay. Thank you, Mr. Kishan, for the question. Next, yes, Bhupendra. Mr. Bhupendra, you can continue with your query.
Thank you, ma'am, for the opportunity. And thank you, sir. Well presented your work. Although I was very late to join you, I was very interested to listen you, but unfortunately, due to some reasons, I was not able to join you. Yeah. So my question is that how do you think of that the CRISPR system for the bacterial system, more importantly for the lactococcus bacteria, because lactococci is one of the bacterium for which engineering is very difficult. So how do you look at that the system, CRISPR based system can be developed for the lactococci?
Okay, thank you, Mr. Bhupendra, for this nice question. Again, you're touching my kryptonite because, as I said, we don't have a big expertise in bacteria models. Our expertise goes more into mammalian models. So we do work with different systems. For bacteria, we only have some vectors. So honestly speaking, this is not our strong suit. If you miss a little bit of the presentation, Anisha and the team can provide it for you after the meeting, for sure.
Thank you very much, sir.
So, just one more question we have received in chat. So, what are the most critical validation step when moving a CRISPR engineered cell model from in vitro to in vivo? So,
well, that's a great question. So basically, when you go into in vivo models, comparing to in vitro, you're going to have a lot of barriers that is going to block your system, because basically when you jump in vivo, first of all, if you use a lentivector, take into consideration that the in vivo model is going to contain your target cell lines plus other different type of cells that can be also able to get infected. So you're not going to control 100% that you're going to be modifying only one target cell line unless that you do some modification. So normally when people go in vitro, they use standard promoters like CMV promoter. So that's something that we change when we jump into in vivo model. So let's say that we want to only transduce some hepatocytes or some given hepatocytes. So we change the promoter. In that way is going to be hepatocyte specific. Second is going to be the titer, because normally when you go in vitro, the titer is more straightforward and lower and easier to manage. But when you go in vivo, again, you're going to have some natural barriers that are going to decrease tremendously the efficiency of your delivery system. And third, it's the main question, is depending on the theoretical approach or your kind of design, is if you want to make a stable cell line generation or if you just want to have a transient application. So some people jump from doing a stable generation with lentivectors and then jump in vivo with an AAV model, not a lentivector anymore. First of all, because AAVs can be tissue specific, because the serotypes are more driven to go into some specific tissues or some specific cells. And secondly, because the effect of the AAV is going to fade away as they do not integrate to the host genome. So that's basically the main variable that we have. Again, summarizing, the promoters can be a little bit limiting or too generous to transduce so many different types of cells. second, the titer, and third, the different stages in which you want to affect, if it's going to be stable or if it's going to remain only for a while.
Yeah. So, okay. So, okay. Thank you for the response. Okay. We don't have any more queries. I think they will reach us out as per their detailed explanation and all. So on behalf
Terrific.
of
Before I leave,
Christian,
Anisha, I have a question because I'm not very well in your language. So how do you pronounce this dish?
it is khichdi, as being an
Khichdi,
Indian.
okay.
Khichdi. Also,
Okay.
it's a kind of khichdi, you can pronounce.
Okay, terrific. Thank you.
Okay. On behalf of Christian Biosystems and ABM, thank you once again for your participation. We look forward to welcoming you to our future webinars. Have a wonderful day ahead. Thank you once again. Thank you.
Right. Thank you, everyone. Bye.